Hi, everyone. My name is Zhang Li. I'm a postdoc from MIT. And first, I would like to thank the organizer, Dr. Gross, giving me this opportunity to present my research here. And today, I will talk about the mechanics of diseased red blood cells. And I will focus on the interactions between the lipid bilayer and the cytoskeleton. Here is my outline. First, I will make a brief introduction of the problem and show you the motivations. Then I will describe our two-component red blood cell model. Then we model the lipid bilayer and the cytoskeleton and two different layers. Then we will use this model to study three cases, including malaria-infected red blood cells, sickle cell disease, and the hereditary spherocytosis. Here is a red blood cell. When it at rest, it has a beautiful biconcave shape. For me, it's like some flat donut. We think the red blood cell is simple. Actually, personally, I think it's not. Because, for example, we even don't understand why it has such beautiful biconcave shape. However, we know the cell is only a membrane like increased time hemoglobin solutions. The membrane consists of two layers. One is a lipid bilayer, shown here is a liquid. The other layer is a cytoskeleton triangle network, a protein network. And these two layers connect together through some transmembrane proteins. And this interaction, they can slide in against each other or some pulling. This interaction between the lipid bilayer and the cytoskeleton play a significant role in the shape and the deformability of the red cell. Although, if something mistake happen in these proteins, mutations will cause a lot of diseases. So that's why in my presentation, I will focus on the interaction between the lipid bilayer and the cytoskeleton and model them and two different layers. The most important motivation to study red blood cell is that there are more than 1 billion people suffer from red blood cell diseases. For example, malaria, every year they kill 1 million people in Africa, cause a lot of tragedies. The malaria parasite will make red blood cell more stiffer and more sticky. The second example I will show is sickle cell disease. I don't want to talk too much because a lot of speakers also covered this. The third example I will talk about is hereditary photocatalysis. In these diseases, a protein mutation in the membrane then makes the cell become from biconcave shape to a spherical shape, then become much more rigid. Then when they pass the spleen, they cannot pass, then the number of cells decrease, they will cause anemia. Now, the main function of our spleen is to filter the blood. Here is a very special, there are some very special blood vessels in the spleen. You can see these lines are the endothelial cells. There are some very narrow openings between these cells. Then all the red blood cells need to pass through this endothelial cell slit. However, if the cell became abnormal, like stiff or sticky, then they cannot pass through this endothelial cell slit. They will get stuck, then eaten by the microphage. Then the number of the cell decrease, causing anemia for sickle cell disease, maybe, or for hereditary spherocytosis. But this is good for malaria. Malaria also causes the cell become stiffer, but if they can be cleaned by this mechanical filtering process. However, unfortunately, some malaria cells can still pass through this slit, then get into our blood, then transmit to another person. Anyway, what I'm saying is that this mechanical filtering process is very critical for red blood cell mechanics for these several diseases. Here is an image from experiment showing this endothelial cell slit. You can see this is a red blood cell. This is the endothelial cell slit. Our collaborators from France, they get the spleen from some patients because some patients need to get cut the spleen. Then they do an ex vivo perfusion. By doing that, they use TE and they can measure the exact geometry of this endothelial cell slit. So based on this information, we can do an accurate modeling. And that's the slit length average. Then, however, in the experiment, it's hard to measure like the exactly cell deformation and even the pressure. That's why we need our simulations. This is a another like SEM image of this spleen slit, this red blood cell. Now I will describe our two-component red blood cell models. Basically, we model the lipid bilayer and the cytoskeleton as two different layers, but we can implement in continuum model and like dissipative particle model. I will talk about the continuum model first. Actually, our continuum model is also a multi-scale model. Basically, we have 3 levels of models at different length scale. Then we couple them together by using multi-scale method. For example, we have a model of spectrum. Spectrum is like the red cables in this triangular network. We modeled that nonlinear spring, but it also has some folded domains. When the stretching force is very large, then the domain may unfold, then release the force. There is a self-protection mechanism. We use Monte Carlo method to simulate that. After we get this stress curve, then we can pass then to our next level model is a junctional complex model. Junctional complex is a basic unit in this protein network. Actually, we get the exact geometry of this unit by studying the binding site between the actin and the spectrum. We build this model. For example, the blue cylinder is the actin. The solid lines are spectrums. We know the exact binding site and geometry of this unit. Then this is a Brownian dynamic simulation of the thermal fluctuation. Also, we can simulate how we can stretch this unit. Then we get the stiffness of this unit. After getting the information, we can pass them to our next level model is a wholesale finite element model. In this model, we model lipid bilayer and cytoskeleton as 2 different layers. Lipid bilayer, 0 shear stiffness, but large area and bending stiffness. Cytoskeleton, there's no big shear stiffness, but no bending stiffness. Now, I will show you a very interesting experiment to illustrate our model. It's a micropipette aspiration by our collaborator in UC San Diego. You can see they apply a pressure, then the cell will get through this micropipette. Please pay attention. When the pressure reaches some value, there's the necking happen in the middle of the session. A portion of the cell detached from the cytoskeleton. Using fluorescence, they find that there's no cytoskeleton inside that vesicle. It means the bilayer detached from the cytoskeleton. Then we can simulate that using our 2-component continuum model. The pink layer is the cytoskeleton. The red layer is the lipid bilayer. We predict that the maximum interaction force happening in the tip, then the separation, but indeed, the necking happening in the middle of the session. We also found that, as I said before, the spectrum unfolding is a self-protective mechanism. But here, actually, if we increase the spectrum unfolding, it's increase the chance of its rupture because you can see when the unfolding happens, then the area of the cytoskeleton dilates. Then in normal direction, the force applied on a single junctional complex increased. We also implemented this 2-component model recently in a dissipative particle dynamics. There's several advantages. For example, the first one, we can incorporate the polymerization of the hemoglobin. This is a snapshot of our simulation to see how the sickle hemoglobin push the lipid bilayer and is an uncoupling between these 2 layers. And also, another advantage is we can incorporate the thermal fluctuation. That is hard for continuum model. Now, that's our models. Now, we will apply this model to study 3 cases. Malaria first. The malaria we study here actually is sexual stage of malaria. It's called gametocyte. Then the parasite became female and male. And to simulate that, first we need to get the exact geometries. Then we use a 3D confocal microscope and use different fluorescent proteins to mark the parasite and the membrane. Then we can get the exact geometries of the parasites in different stages. That's the geometry. Then we need the stiffness. How to measure the stiffness of the parasite inside the cell is a challenging problem because you cannot break the membrane. It's hard to do that. But we found that there's an interesting, good way to do that. When we aspirate the cell from the head, we found both the membrane and the parasite will get into the pipette. However, if aspirates the cell from the side, usually only the membrane will get to the pipette. So we simulate that using our continuum model. We model the parasite as an elastic body. It's like storing a two equations with two variables, then we can get the stiffness of both the membrane and the parasite for the first time. After we get the geometry of the cell and the stiffness, then we also, as I showed you from our collaborator, we also know the exact geometry of the spleen slit. Then we can simulate how the cell passes the spleen. What's the critical pressure for this cell to pass the spleen? This is only the first example. We have like different stages. You can see during the development, the parasite is a red one. They start growing, but they change the shape. Based on our measurement, the stiffness of the membrane, the parasite, like increase from stage 1 to stage 4, but they sudden drop in stage 5. Then after we get the critical pressure is y-axis. The x-axis is the spleen slit width. We find an interesting result. For the earlier stage cells, like from 2 to 4, the critical pressure is very high than the mature stage 5. It means it's only possible the mature stage the critical pressure is below the physiological pressure. Actually, that is true. It's consistent with clinical observations because the doctors, they only found the mature stage cell in our circulating blood. Then that's bad because if there is a cell in our circulating blood parasite, then we'll pick up by a mosquito, then maybe transmit to another person. So this result, in spite if we can develop some kind of drug to control or manipulate of the stiffness of the cell. Actually, that's true. We are collaborating with Novartis. Using this drug, we are doing the micropipette aspiration again. We found that this drug indeed changed the stiffness of the parasites. That's the story of the malaria. Now I will go to the sickle cell disease. For sickle cell disease, instead of using micropipette aspiration, we use another technique called diffraction phase microscope. Basically, we shed a laser beam through the red blood cell. After the laser passes through the red cell, the phase will shift. Then we compare the phase with the original phase. Then we get the thickness of the red blood cell. During time domain, then we get the fluctuation of the cell membrane. Then this is the fluctuation and the thickness of a healthy cell and the different sickle cell disease, type 1, type 2 to type 4. After we get this experiment data, the question is how we can extract the mechanical properties for different cells. We have an analytical model, but it's only for a spherical cell. That's not accurate. That's why we can use our dissipative particle dynamics to simulate how the cell vibrates. Then we can extract like the bending stiffness, shear modulus, and area modulus, even the viscosity inside the cell. We published this active biomaterial, and we find that even compared to the one component the DPT model, the two component model has advantage. The y direction is the amplitude of the fluctuation, and for different cells, we found the two component model, the showed you model the bilayer and the cytoskeleton as two different layers. Actually, the result is more close to the experiment. After get this mechanical property of sickle cell, then we are doing microfluid experiment of sickle cell disease. We have like multiple obstacles in the microfluid channel. Then we can even measure how fast these cells pass through this microfluid channel. Now I will show you a movie of the experiment, but it's for healthy cells. You can see this is a red blood cell, some moving faster, but some moving slower. Based on the velocity, we can to compare what's the stiffness. Then we can control, we now are control the oxygen level, then to study how fast they can pass, or even whether they can pass or not. This is a experiment. Then I'll also do a simulation, like how the cell passes this. You can see two colors because it's a two-component model, lipid bilayer and the cytoskeleton. That's the example of sickle cell. Now, finally, I will also talk about the hereditary photocatalysis. In these diseases, actually, it's not a single reason. For sickle cell, it's a single reason because there's a mutation of the hemoglobin. But for hereditary photocatalysis, there are many reasons. However, there are only two hypotheses. The first hypothesis is that during the cell motion, there may be the lipid bilayer, the bond between the lipid bilayer and the cytoskeleton may break. So this part of lipid bilayer will buckle and cause the membrane loss. When the membrane loss, then the cell surface area will decrease, but the cell volume are almost the same. Imagine which shape maximizes the volume to surface area? It's a sphere. That's why it's gradually became a sphere. Then much richer. You can see if it's a basketball, it's very rigid. If you want to make it softer, you pour out of the air. That's the hypothesis 1. Hypothesis 2 is that there's no break of the bond, but there's some vacancy in the cytoskeleton, the deficiency in the cytoskeleton, a big hole. Then the above lipid bilayer became unstable. Then the buckle then caused the membrane loss. Now, using our two-component model, we can quantitatively study these two hypotheses. For example, for first hypothesis, we try to measure the strength when the bilayer skeleton bond break. For that, we duplicate an experiment done by Professor Hodgmuth from Duke. In this experiment, they touch the cell to a substrate. They coating the substrate first. When then sometimes they touch to 2 points, sometimes they touch to 1 point. They apply shear flow. The interesting result, they found that at a critical shear stress like this value, then the tether will form. The tether is indication of the separation between the lipid bilayer and the cytoskeleton. So using our two components, we can simulate this experiment. For example, in the first plot is a simulation of a 2-point attachment. X direction is shear stress, Y direction is deformation. This is our simulation result. It's a validation of our model first. Then we simulate a single attachment case. You can see, then in this region, the bilayer skeleton interaction force is very high. The counter is the force, maximum force applied on single junctional complex. We estimate it's a 10 piconewton per junctional complex when the bilayer cytoskeleton starts to separate. This is also consistent with the adhesion energy theory developed by Richard Waugh, but their theory is a continuous theory. But in our simulation, we model that the real number of the spectrums is more realistic. This is hypothesis 1. In the second hypothesis, as I said, there's no bilayer skeleton bond break, but the skeleton may be expanded too much. Where it may happen, I think the two possibilities. One is in our heart, there's a strong shear flow, or the other case is in the spleen. Now, let's consider the first case first. We simulate in the shear flow, the relative cell will do a titrating motion. We coupled with our previous two-component model, we developed a boundary integral method that simulates the fluid. Then you can see the brown layer is the lipid bilayer, the light blue layer is the cytoskeleton. I attach 2 beads on both layers. You can see during titrating, these 2 beads stick together, overlapped. However, in spherocytosis, people find that the band 3 density is only 10% of the healthy cell. What caused the friction between the lipid bilayer and the cytoskeleton? We can imagine when the bilayer skeleton starts sliding, actually the skeleton transmembrane protein is inside the lipid bilayer. Lipid bilayer is a liquid. Then it's kind of drag of this protein moving in the lipid bilayer. Then the friction is caused by the summation of this drag force of these proteins. If the band 3 density is smaller, then the drag coefficient should be smaller. When we decrease the drag, the friction coefficient, you find these 2 beads start titrating, but there's some slip between the lipid bilayer and cytoskeleton. That is very important. Why? Because lipid bilayer is area incompressible. If these 2 layers move always together, then the cytoskeleton is also area incompressible. Then the density of the cytoskeleton never change. However, if there is a sliding between these 2 layers, although the bilayer is area incompressible, but the cytoskeleton may be locally expand significantly. Actually, indeed, there is a decades debate on whether bilayer skeleton slip happen in tank shading. For example, in the recent paper in Biophysics journal, using a numerical model, they found that there is a measurable local area dilation of the cytoskeleton during tank shading. However, 20 years ago, Professor Fish also published in Biophysics journal. The title is the surface area of the red cell membrane skeleton locally conserved? His answer is no. There's no, because the tank shading is too fast, no time for such flow. Who is right? Now, using our model, we can quantitatively study this process. Now, I show you the result. The x direction is time, y direction is the area deformation. I mean, this one is the expansion. It's greater than 1. This one is compression. You can see it takes a very long time for the cytoskeleton deformation to accumulate. This is about like 500 cycles of tank shading. It takes a very long time. However, if we decrease the friction coefficient between these two, it may only take 3 seconds. This accumulation will complete. During that case, you see, in this deformation, then there will be a significant expansion of cytoskeleton during the tip. Then maybe because this situation, there maybe is a vacancy in the tip. Then the bilayer in the tip maybe become unstable. They may lose. Now that's our explanation. Now the second case, I said maybe this bilayer skeleton detachment may happen in the spleen. So we simulate using the DPD how the red cell pass through this spleen slit. We use the geometry we measured from the experiment, and we also study. Actually, the most important result we care here is what's the critical pressure for this cell to pass through this narrow slit. We studied several parameters. For example, the shear modulus, because in malaria, the shear modulus change significantly from healthy cell to ring stage to shy zone. We also studied the cell size, because in our body, there's a distribution of the cell size. Also, the slit width. Finally, we also studied the sphericity. That's how the cell is close to a sphere, because I said in spherocytosis, it's become much more spherical. You can see the critical pressure increased dramatically when it became spherical. Now, currently we are working on, we want to introduce some local deficiency of the cytoskeleton. We want to predict the buckling of the lipid bilayer to predict loss. We also derived an analytical formulation, what's the critical pressure for the cell to pass. But we also want to, using our two-component model, to numerically predict this value in the spleen to compare these two. Finally, since I have still some time, I will tell something about the question I mentioned in the very beginning, why red blood cell has a biconcave shape. Let me show you a recent experiment published in PNS about a tank shading motion at a very low shear. It's like 3 per second. In this tank shading motion, you can see actually the cell keep a biconcave shape instead of an elongated shape I showed you before. Then there's a tank shading. However, using our previous model, we find that under such low shear rate, the cell always tumbling, not tank shading. So it must be something wrong, missing in the model. So we found that in our previous model, we used the biconcave shape as the stress-free shape. I mean, in that shape, there's no stress. But actually, that's maybe not true. So we want to predict we think maybe the original stress-free shape of the red cell is nearly to a sphere, or maybe it's a ellipsoid. For example, predicted by Lin in PS, they found if the cell original stress-free shape is close to a ellipsoid, then it's much easier to predict the biconcave shape by deflate the volume of the cell. Then we do this same thing. The x direction is a spontaneous curvature of the lipid bilayer. The y direction is how it's close to a sphere. You can see when it's very close, when the initial stress-free configuration is very close to sphere, then we always get a cup-like shape, as many people already see. But if we use a ellipsoid, then we can get a nice biconcave shape. This is the area deformation and the shear deformation after diffraction process. It means initially it's not stress-free. The stress. The important thing we found here is that if we use this shape, this not stress-free shape, we can predict the tangentiating motion as I showed you in the previous experiment under very low shear rate. For example, it's here we showed even at shear rate 6, you can see the cell also do tangentiating but keep its biconcave shape instead of an elongated shape. We also find that we can also replace the experiment like the roll. Sometimes depends on the orientation of the cell. It's also do a rolling motion. I want to explain this plot. The y direction, as I said, is how the stress-free configuration is close to a sphere. One is a sphere. We found that if we became close to a sphere, then the critical pressure for the tangentiating to happen significantly dropped. That is consistent with the experiment. However, we don't believe it's exactly a sphere, because if it's exactly a sphere, then there's no shear memory, because people find that. For example, we simulate that we do the tangentiating, but if we stop the flow, then the dimple region will go back to the dimple region. It means the cell has a shape memory. But if the initial configuration is exactly a sphere, then every part of the cell is identical, then there's no shear memory. So based on these results, we prove that the stress-free configuration is like ellipsoid. Here is a summary of my presentation. First, I described our two-component model. We implement both in like finite element continuum model, also DPD model. For malaria, we extract both the membrane stiffness and the parasite stiffness, and we found that only mature stage can pass the spleen. For sickle cell disease, comparing with experiment and modeling, we extract the mechanical properties of the red cell in sickle cell disease. We also apply in microfluidics to study this sickle cell disease. And finally, I also showed you in Hereditary Spherocytosis, we studied these two hypotheses like the bilayer bond break and the bilayer skeleton slip using our two-component model. I have 4 minutes. Then I can also talk a little bit about the future direction. Currently, besides we control the oxygen, we also collaborate with Tony Huang's lab at Penn State. We use acoustic force to manipulate the cell. They put the two IDT together, then apply acoustic radiation force. So because the cells are different size, different stiffness, then the acoustic radiation force on it is different, so that we improve the separation of these cells. Like we can want to separate CTC cells from the normal cell, or even separate white blood cell from the red blood cell. We also simulate this process. For example, this is a trajectory from the experiment of the cell, how it goes. This is our simulations. The second thing we want to do with acoustic is that when we put a cell, like a tumor cell, in between two pressure nodes in the acoustic field, it will stretch it. But how to calibrate, we need the simulation. So we simulate this process. We get acoustic field around the cell. Then we also apply the elasticity of the model. Then we can predict under which kind of power, what's the strain in the cell. So we will do a multi-physics simulation to add the flow and the acoustic and the elasticity together to predict this system. And we also currently build a flexible and active blood vessel using DPD, because as many showed that when the sickle cell releases the hemoglobin, it will consume the nitrogen oxide, then maybe make the blood vessel very narrow. Also, the blood vessel itself is flexible. That's why we are building a flexible vessel model. And also, I showed you in the spleen, actually there's some also stretch fibers inside the endothelial cell is active control this thing. So we are working on if we can build a flexible vessel or this slit and add active control and intact with our red blood cell models. And finally, I want to thank our funding agency, NIH, and also the Singapore MIT Alliance program. I would like to thank my advisor, Dr. Min Dao, and Professor Suresh at MIT, also my co-advisor, Professor Kanyadakis in Brown, and our collaborators, Dr. Pelkin in Switzerland, and Dr. Xuejin Li. He presents his sickle cell work yesterday. Also, Dr. Fedosov, I basically built the two-component model based on his multi-scale DBD models. I also need to thank my PhD advisors, University of California, San Diego. We built the two-component models, and also the Martin's lab they're doing like gametocyte malaria work at Harvard. Finally, I thank you for your attention. I would like to take any questions. Thanks.